couple of weeks ago, I was listening to a documentary titled Human Nature on the CRISPR technology, something that I know very, very little about. And my current guest was prominently featured in it. He was talking about some of the ethics of CRISPR. And so I reached out to him, and he was kind enough to come here to chat with us. Professor Fyodor Urnov, how are you doing, sir?
I'm well, thank you. How are you?
Good. So let me just tell you a bit about this gentleman. He's a professor of genetics, genomics, and development at UC Berkeley. First conference that I ever spoke at or presented my work as a graduate student in 1992 was at UC Berkeley. His research interests include, quote, the development and advancement to the clinic of novel approaches to treat human disease using CRISPR-based genome and epigenome editing. Some of you may know that in 2020, two scientists won the Nobel Prize in Chemistry for their work on the CRISPR technology. So I guess the first question, Fyodor, would be, speak to the layperson, what is CRISPR?
It's a word processor for your DNA. It sounds futuristic, but that future is here. If you were to tell somebody in the 19th, or for that matter, 19th century, for that matter, somebody in the 1950s, that there would be a little box on your desk and you could pull up anything from Moby Dick to a 240-character tweet, and then pushing little buttons in front of you, change it at will, the 19th century, in fact, the 1950s person would ask, what's wrong with you? So when I compare CRISPR to a word processor for DNA, I do this with a due sense of awe. Make no mistake about it. We've never had a technology like this since DNA was discovered to be the genetic material in the 1940s. We've known about the way it comes together since the 1950s. We've been able to read it since the 1970s. And now here we are in 2021, and we can change it relatively at will with extraordinary precision, as if you were clicking with a mouse on a very specific section of text, and extraordinary efficiency. Efficient enough to where, as we'll probably talk in a second, last year, a human being with sickle cell disease had several 100 million of her blood stem cells taken out. A gene edit was performed on them. Essentially, all of them were precisely gene edited, and then they were put back in. And a year plus later, they're all alive and well and appear to have resolved all severe symptoms of her sickle cell disease. So we have moved completely out of prototype, hypothetical dreams on pages of science fiction novels and through early stage proof of concept and all the way to clinical reality. That's what CRISPR is.
Let's drill down further, because while the analogy to a word processor is a great one, it still leaves most people confused as to how does that analogy work exactly. So the human genome is made up of a bunch of discrete, the 4 letters and so on. And we can put how many are there that you could put in the human genome? In the billions, right? What is the number?
Oh, the billions is flat. It doesn't sing. But let me give you a sense of the scale of the human genome in more human terms. Imagine you had a textbook to read it, the whole thing. First of all, you need 500 textbooks. That's how long the genome is. And now imagine you start reading it one letter at a time. A, G, C, A. It would take you a century to read the entire human genome one letter at a time. It's 6.6 billion letters. But again, it's a bit like saying the Earth has 7+ billion people. It's a number that's so large it's hard to grasp. Yep, it would take you a century to read the entire human genome.
So now you've got this whole sequence, however many pages it is. Mm-hmm. Now, when you say CRISPR goes in and edits a particular section, so the first premise there would be that I know unequivocally that the place in the genome that codes for sickle cell anemia is exactly that and nowhere else, so that I can target that. So that would be the first thing that I would have to know, right? But for example, personality traits, it's a lot more difficult for me to know where assertiveness is coded in the genome, right? It could be in many different places. Many different genes could be involved there. So is the technology of CRISPR restricted to cases where I absolutely know the exact location of where to go in the word processing document to edit those particular letters?
with great power comes great responsibility, right? I forget if this is Spider-Man or something more philosophically deep. The magnificence of Jennifer Doudna's here at UC Berkeley and Emmanuel Charpentier, Nobel Prize winning discovery of how CRISPR works, is that technology is agnostic to the editing that it drives. In other words, you can make a change whichever one you want. Question is, what are you doing with it? So the moment we step away from the very narrow focus on how CRISPR itself arrives inside a human or a cow or a rice cell, and CRISPR has successfully worked in all of those settings, and we have routed CRISPR using the Doudna-Charpentier Nobel Prize winning discovery rules to gene number 1 or gene number 57, with gene number 1 maybe being something that causes severe disease and gene number 57 something to do with personality. At that point, the technology in the narrow sets kinds of stops, and what begins is the human element. Why are we doing the editing? What outcome are we expecting from it? And to this, we have to bring not only, the technology progress of the past decade, but, 35,000 years of development of human civilization, morals, ethics. And I can tell you that all of us in the field, every single person who has been working on gene editing for the past, quarter century that it's been in the works, are frankly mortified at the notion that the fruit of our joint labor will be used for how do I put this, quixotic or Icarus-like applications where people try to fly to the sun using wings of wax. Our primary focus, because it's new technology and its clinical track record, or in fact its track record in the real world of agriculture, for example, where its promise is enormous. Just to give you a sense of how significant the ag promise is, the Innovative Genomics Institute, where I have the honor of working, that Jennifer Doudna founded, its mission is to use CRISPR to improve public health. And what are we doing? Well, of course, we're using CRISPR to try to treat disease, but an equal size effort is to use CRISPR in agriculture, because we all believe that making the next generation of crops to feed a growing and warming planet will frankly have a bigger impact on humanity in the next 50 years than, being able to use CRISPR to treat disease number 4. Don't get me wrong, we're passionate about treating disease. So back to what we're doing with this technology. There's a big difference between saying, here's a specific disease, sickle cell disease, which causes severe pain, or congenital blindness, which needs no explanation, or taking something like, rice dying of high temperature or of severe disease. So in those settings, you go in with CRISPR, you make the desired change and expect a very specific outcome. No more pain. You're able to see now. The rice plant is able to tolerate a higher temperature due to global warming, or is able to tolerate being attacked by some terrible disease. And all the way at the other end, at this point we smoothly transition into, Brave New World and notions of, genetically segregated groups of humans that have been engineered in one way or another. All of us right now are very focused on the former. How do we do relatively, quote, simple, unquote, edits to humans or crops or animals with very specific goals for improvement of the planet, and are frankly shying away as best as we can from notions that we would take this technology anywhere beyond that for now. Now, this doesn't mean, every technology initially has an application and suddenly becomes relevant in entirely new settings. And there's no question in my mind that in the long term, CRISPR gene editing will find its applications in realms of human existence that we currently can't even imagine. And in fact, for recently, work from Jennifer's group and other groups has produced a way to use CRISPR in something completely different, which is CRISPR as diagnostics. And in fact, again, here at the Innovative Genomics Institute, we have a lab to test for the virus, and we're actively exploring ways to use CRISPR to test for SARS-CoV-2, because it could be faster, it could be more sensitive, and we're not alone in doing this kind of work. So I can't really predict which way this is going to go, but I'm super hopeful that the next, let's say, 5 to 10 years will be exclusively focused on severe disease, save the planet, that kind of stuff, rather than, and now Let's try to write anti-utopias and cast films with, Ethan Hawke in Gattaca Part 2.
Right. Again, forgive me if any of the questions are too basic, but remember that our audience are made of very intelligent people. Many times they may be lay to the topic that we're discussing. Does a CRISPR intervention work both on somatic cells and germ cells? Can you make an intervention on person A, and then that is passed on, or I guess that's a Lamarckian type of question, or is it when you circumcise Jewish boys, their sons are not born circumcised, right? So there is no theory of acquired trait, right? There is no Lamarckianism when it comes to at least the most basic of our understanding of evolutionary theory. So how does the CRISPR intervention work? Can it work on both cells, or is it largely within the confines of that person's lifespan?
You are touching on one of the most profound and, let me be clear,
unbeknownst to me,
unsolved right now by all of us challenges in the future of CRISPR. I love the way you phrased it in terms of circumcision, because the way you did so evokes one of the most distinguished scientists in the history of my field, which is genetics. His name was August Theodor Weismann, and he is one of the founding fathers of genetics for a very profound discovery. And that is that the body of a living thing, in fact, is two separate things. It's the body, which walks around or grows, and then it is what Weismann called the germ plasm. It's a really awkward term. What he meant was the reproductive system, the part of the body that produces the next body. And just like you, Weismann has argued that millennia of various practices, such as circumcision by observant Jews, or, for example, foot binding in certain parts of Asia, have not produced newborns that are circumcised, nor have produced children with smaller feet. Right. And of course, once this was connected, and this didn't actually happen until much later in the 20th century, with an understanding of how Mendel's laws, discovered in the mid-1860s, and Darwin's notions of how living beings arise by natural selection, all of this came together to basically repudiate the entertaining but erroneous visions that Lamarck had for all of us, which is, a giraffe grows taller because it reaches its neck to grab a fruit, and then its body somehow communicates the need to have a longer neck to its offspring. That doesn't happen. It just doesn't. Until CRISPR.
Right.
It is absolutely possible, in principle, to use CRISPR to genetically engineer a human being's, quote, germ plasm, or in English, reproductive system, to have that genetic change transmit to their progeny. Now, right now, in very practical terms, if you wanted to do that, and I really, really hope nobody listening to this wants to do this because it's neither safe nor ethical, you would probably do what a tragic and painful figure in the history of humankind, frankly, a person named Jianhui He allegedly did when he took human embryos and injected them with CRISPR, and then in an act of breathtaking, ill-advised hubris meets Napoleonic gall. I don't even know what negative turn of phrase to use here. His effort led to the birth, it is alleged, of two children. We're all, our deepest heart's wishes are that the two children are safe and well. This should never be done again, in my personal opinion. But in practical terms, it can be done. And so when you ask me, can CRISPR breach the barrier that existed unbroken until now between the body and what happens to it, and how whatever happens to the body is passed on to future generations? Yes, CRISPR can breach that boundary. And wow. I am deathly apprehensive.
So, I wonder if you're familiar. Are you familiar with the Dutch famine studies?
I deeply familiar, and I've been teaching it in my classes.
Oh, there you go. So those studies seem to be suggesting that Lamarck could be turning in his grave saying, "Hey, I told you so." Yes, maybe it may not be relevant to explain the evolution of a morphological trait like the giraffe's, the size of its neck, but those studies certainly point to the fact that there's something that's captured in what is now referred to as the epigenome, right? So is CRISPR straddling that discussion? I mean, I guess so.
Yes,
I guess from
it
okay,
is.
exactly.
And in fact, and this again is a work from Jennifer Doudna and Jonathan Weissman at UCSF. CRISPR can change the epigenome as well. And now to take a short step back to not mortify your audience with yet another term, genetics is DNA, right? A, C, G, T. We have genes. We have about 20,000 of them. If you look to your left or to your right at your fellow human being, there is a single genetic letter different about every 1,000 letters or so. And those differences explain some fraction of why we're different.
Which, by the way, before you go on, let me interrupt you for a second, Fyodor. In a sense, it is a very liberating message because it demonstrates that we are so much more alike than different, right? I mean, if people truly understood genetics, they would understand how astonishingly similar we are, notwithstanding our differences in skin color and height and so on, right? I mean, in that sense, it's a kind of a socially justice congruent discipline, right?
I know this is two professors at distinguished research universities basking in the glory of our egalitarian worldview, but yes, absolutely. I should also point out and honor the memory of a great scientist, Richard Lewontin. He was a professor at Harvard. And in fact, the data that you are alluding to and the implication of those data for, the fundamental kinship of humanity and a focus on that kinship rather than Huxley like, oh, I'm an alpha, I'm so glad I'm not a beta. And all of that nonsense. Richard Lewontin is one of the key figures in the history of 20th century biology to draw first attention to that point, which subsequently has been supported by 50 years of detailed studies. So yes, it's absolutely true that we are highly genetically kin. You look to the left, you look to the right. Every 1,000 letters, there's a single difference. Not that different. But then, where things get nuanced, and let me emphasize that this is interesting nuance. In other words, this is not detail for purposes of details, because of course, as scientists love to get lost in the details and lose everyone else in the process. Our genes, the DNA we get, they learn from experience. So as we are born, or in fact, as we're in our mother's womb, and as we are born, every bit of exposure to the world of every flavor, this could be what mom ate, this could be how we were delivered from mom's womb. What did we eat? The world, how did the world around us? Our genes remember. And how do the genes remember? Well, we are used to the notion that some environmental exposures can actually change DNA. And this is why you have to wear sunscreen, because your sunlight will change your DNA, and those changes will cause cancer. This is why it is highly advised to not smoke tobacco, because tobacco has chemicals that will go into the DNA of your lungs and change it and cause cancer. But the majority of exposures that we have to the world don't really change our DNA in any substantial way. But what does happen is our genes listen to the world. And I'm using highly anthropomorphic language. I will switch to molecular in a second. And what changes over our lifespan is, if you will, a sort of makeup. If you think about sort of the concept of makeup and how humans since the beginning of time have been applying things to their exterior in order to change the way they present themselves to the world. So DNA acquires makeup as well. And it's not magic. It's not, pixie dust or made of mysterious material. We know what the biology is. The biology is there are specific chemical marks that are put on the DNA. Now, they don't change the letters of the DNA, but they change what the DNA is interpreted as saying. So, for example, let me give you a public health relevant specific. Folks, eat a lot of fiber. Why? Because it will keep your colon healthy. Why? Because the fiber ferments in the gut, and the products of that fermentation protect the DNA in your gut from losing the ability to fight cancer. So the DNA stays the same, but if you eat a lot of fiber, then the fermentation byproducts will keep the makeup, the epigenome of the cell, of the DNA in your cells, healthier. And it's just an experimentally established fact. So CRISPR can be used to change DNA. So you go into a cell and you change one letter to another. CRISPR can be used to change the epigenome as well. Namely, rewrite the molecular makeup, the sort of the eyebrow liner or the lipstick or the what have you, or the body art, over certain stretches of DNA. And some of this will last. Now, before we envisage a future where everything goes according to Lamarck and people use CRISPR to epigenome themselves and their children, I want to emphasize that this is still a matter of much research and, frankly, uncertainty. It is unequivocally the case, and I think that the horror of World War II is just, the worst tragedy in the history of humanity. But out of tragedy came the profound learning that exposure of a fetus in utero to a particular set of environmental stimuli that only mom experiences carries through into the newborn child, into the adult state of that child, and appears to then carry through into the children of that person. So these are grandchildren of women who suffered in World War II. What's happening there? Has their DNA changed? No. The implication is something in the epigenetics has been altered. I want to emphasize that to what extent these kinds of phenomena are common, again, huge amount of uncertainty. But in principle, does this chart out a concept where CRISPR-based epigenome changes could be heritable? It does, I hate to say. Again,
wow.
But I want to caution against the notion, oh my goodness, what have we unleashed? This is early technology. The overwhelming majority of its applications are to treat genetic disease, infectious disease such as HIV and potentially COVID-19, to treat cancer such as leukemia. And I am emphasizing that all of this is actually happening. This is not a headline in a journal of the future. This is an entry on clinicaltrials.gov. So there's a big difference, right? But I think now is the time in the history of our species to ask ourselves, okay, we've acquired Google Docs for our DNA. Now what?
What are the ethical guidelines, if there are any so far, in terms of what CRISPR can be used for versus what it can't or shouldn't be used for? So, for example, okay, let's solve sickle cell anemia. Fine, great. Let's change the baby's eye color to be as beautiful as those of Professor Saad, a beautiful green. Maybe we shouldn't do that. That's too vain. So is there a set of guidelines, bioethical guidelines that, the great people who are working in this field have come up with, or is this still a work in progress? Where are we?
No, this is actually, I'm very happy to speak to this because a key part of the effort here at the Innovative Genomics Institute is to drive forward ethical applications of CRISPR while doing what we can to communicate to the general public what we should stay away from. So what's ethical right now? For human beings, what is ethical is the use of CRISPR to treat existing severe disease. And let me walk through there. There's actually, there's more than one key point in that simple sentence. First of all, let's talk about the word existing. Right now, the only type of clinical trial in the United States and in Europe that has been authorized by the Food and Drug Administration stateside or the European Medicines Agency in Europe is to treat human beings who are currently gravely ill. And I'll give you specifics. Sickle cell disease is a grave condition. A related disease of the blood, of red blood cell production, thalassemia, is grave. And people with it have been treated with CRISPR. Congenital blindness is devastating. A person, in fact, probably several, have been treated with CRISPR. Leukemia is existing and devastating. Again, there has been a CRISPR trial. Most recently, we've heard the remarkable news that the biotechnology company Intellia became the first one to inject CRISPR into the bloodstream of 6 individuals who have a severe existing genetic disease which harms their nervous system and their heart. And so, right now, whether you show up in front of the FDA in the US, EMA in Europe, Health Canada in Canada, or the Therapeutics Good Administration in Australia, or any such regulator, the only path forward is if you say to the regulators, we would like to take adult or pediatric subjects with existing severe condition, and we would like to CRISPR them. Any application other than existing disease will be not allowed. Then the other word is the word severe. Some things are chronic. Let's say rheumatoid arthritis, it's devastating. It's inflammatory bowel disease. I just named 2 autoimmune conditions. It's terrible. Multiple sclerosis is profoundly painful. How about migraines? They are terrible. I received a letter recently from a human being with a severe migraine, and they wrote to me about what it's like for them to be who they are and to carry the horrific burden of being incapacitated by this awfulness. So we have to be incredibly careful, and it is in fact a very firm regulatory stance of the FDA in the US and the EMA in Europe that the disease has to be severe, and this is because we don't know what the long-term safety consequences will be of CRISPRing anyone for anything. And so we have to be incredibly mindful of complying with the laws of protection of human subjects in research. And there, the central principle is the principle, again, an eloquent, a single word that speaks very eloquently to all of us, the principle of beneficence. And it means you must maximize the good. You have to go above and beyond your call of duty to do good and to minimize harm. And the principle of beneficence argues that we should only treat really severe disease. And that doesn't mean that rheumatoid arthritis or inflammatory bowel disease are not severe, but there's a difference between those chronic conditions, which are currently somewhat managed by medication, and, leukemia, which is about to kill somebody in 4 months.
Right. How do we, before you go on, how do we gauge, since you refer to the long-term effects of a CRISPR intervention, how do we establish that, right? I mean, we can go in, change something in the genome so that the person who suffers from sickle cell anemia is alleviated of that burden or that condition, but then through, for example, a pleiotropic effect, a downstream effect that it can affect a different set of genes. How do we know that you changing the word processing document at position X doesn't have some downstream effect, anywhere else? How do we establish that?
You just used the word that I wished was part of the common vernacular and not just subject of, genetics professors standing in front of their upper division undergraduate class.
And evolutionary psychologists who know a lot.
Oh, for sure. Pleiotropic is Greek for in many places. It means a single change that has multiple consequences. And we don't have to go far. Sickle cell disease is caused by a single letter. I talked about having the human genome takes a century to read. Imagine you're reading 2 such century-long books, and one of them you make one mistake once in that century. And if that mistake happens in a specific gene, one called HBB1, or hemoglobin beta encoding subunit, you're in trouble because you're going to get sickle cell disease, and its single genetic change will cause acute episodes of pain, susceptibility to stroke, susceptibility to pulmonary complications, a vascular necrosis, which basically means your joints are severely, irreparably damaged, kidney damage, and the list goes on and on and on. So a single genetic change, multiple consequences, pleiotropic effect. By the same token, I'll give you a specific. We don't have to go far. Across the bay at UCSF, scientists have discovered that a single genetic change in a gene called DEC2, which doesn't say anything, has a remarkable effect. People can sleep 4 hours a day and be just fine. So, 64,000 letters of genetic code question. Should we be giving everybody that genetic change? I certainly would sign up for a life where I could sleep only 4 hours. And the problem is we don't know if that change is pleiotropic. It's very difficult to find out if, we give people the short sleep state and what else will happen to them.
Well, is it almost, combinatorially, is it not pretty much intractable to ever be able to answer that question?
Well, okay. So it really depends on whom you're speaking with. Folks of my persuasion, which is, and I want to be clear, I'm talking about 20,000 people and more. In other words, scientists who are using CRISPR to try to improve human health, argue that, if we're working on something legitimate in the name of betterment of humankind, then we should seriously consider it. Now, giving people short sleep is currently, not high on our agenda. But if you want to make a change that we think would help people, then there are FDA-prescribed experiments you have to do on animals of various sizes. You start with mice, and then you proceed to monkeys, and see what happens. And I'll give you a specific from yesterday's headlines. There is a biotechnology company called Verve, V-E-R-V-E, there in Cambridge, Massachusetts. And they've recently described a remarkable set of studies where they've used a new flavor of CRISPR called base editing, developed by David Liu at the Broad Institute, to change one gene. Now, the gene has a boring name, PCSK9, but what's really interesting about it is very rare people who have a different form of that gene are more than 80-fold protected from heart attacks.
Wow.
I know. And, as you can tell from my name and accent, I am Russian by ancestry. And, the Russian national breakfast is eggs fried in butter. Not exactly the American Heart Association specified vision.
Although we don't know anymore, because now I hear some cardiologists saying that it's completely false to worry about eating eggs. So
can I talk to those cardiologists? Because I love that kind of food. But so imagine a setting where we tell people, you know what, enjoy the third croissant with an extra side of lard. And because we've just CRISPR'd you and we've given you this heart disease protective gene. So this is not science fiction. There is a biotech company run by extremely sophisticated and accomplished physicians and scientists who is going to actually do that for people at major risk for heart disease. But the reason I brought that up in answer to your question, can we ever know whether a change is safe? Well, complete safety can only be established from an extended track record. And as the COVID experience has shown, some rare safety signatures can only be seen after you've treated millions of people. The AstraZeneca vaccine is associated with clots, but we've found that out after, tens of millions of individuals have received the vaccine, right? So in this case, the regulatory standards, so what the Ministry of the Food and Drug Administration or the EMA will tell you, go do this experiment on non-human primates, which is monkeys, and see what happens. Wait, look, make sure that the animal is okay. Make sure that the desired effect is there. Now, a monkey is not a human, but experimentation on humans is called a clinical trial. And you're not allowed to do a clinical trial unless there's a very strong justified need. And in this case, it's severe disease. So to be clear, I want to emphasize that what Verve is trying to do right now, and other companies doing similar things in these kinds of disease areas, they're not proposing to take, a Berkeley professor of Russian ancestry who is currently healthy, I hope, and CRISPR him so that he can enjoy his morning eggs and butter. No, that is wrong. That is not what to do. They're proposing to treat people with severe genetic forms of heart disease, lipid metabolism, which causes severe life-threatening risk, and try to protect them. And if that were to be safe and effective, okay, well, then we can talk about using this for disease prevention. And, many in your audience, I imagine, are taking a statin to, keep their blood chemistry healthy. But most folks would be surprised to know that statins were not developed initially to prevent heart disease. They were developed in the 1980s to treat severe existing genetic heart disease. And when they were used, they were shown to be safe and effective. And then clinicians and scientists who were looking at the data went, wow, does this mean we should be using it for sporadic heart disease that's not severe and genetic? And that was done, and that looked great. And then gradually, as this treatment moved from being able to help rare folks with genetic conditions to then being applied to a broader group of folks with existing disease, the regulatory authorities around the world have said, fine, let's just allow physicians to prescribe a statin for heart disease prevention. That is the vision. I don't know if that will happen for CRISPR, but I hope it does, because again, butter is tasty, but not good for you.
And butter is tasty, and we're attracted to it for evolutionary reasons, right? We've evolved our gustatory preferences in an environment of caloric scarcity and caloric uncertainty. So it only makes perfect adaptive sense for us to want that extra piece of buttered.
You and I could have a fantastic conversation about this. I've had the good fortune of traveling only a little bit in Asia, but I can tell you that after a week in Japan, or a week in, let's say, Guangzhou, and I've done both, there's just not a lot of butter around. There's just like and there's not a lot of cheese, and I thought I could not live without cheese, and then 3 days of attending a conference in Kyoto, suddenly the broccoli and tofu seem really yummy. So it's kind of it would be fun to ask. Don't get me wrong. There's no question that genetic differences track with diet, and the two greatest examples, of course, is milk. Milk-producing animals have been domesticated at least 3 times in the history of civilization, in the Middle East, in Europe, and in certain parts of Africa where pastoralist agriculture is practiced. And the domestication of the animals has led to the genetic evolution of humans to be able to...
I actually lecture about this. It's called, as you know, gene culture coevolution modeling, or dual inheritance modeling. And so I precisely use the lactose intolerance story when explaining that model. So great, yes, I'm with you.
I'm not. There's no doubt. If we carefully genotype a bunch of Russian and French people, maybe we will see that we love butter genetic change. I don't know if that exists, but talk about epigenetics versus genetics. Our genes and our minds learn from experience. I've lived in Berkeley for 21 years, and extra virgin olive oil is pretty good. And I didn't, believe you me, I did not grow up on extra virgin olive anything.
Well, I'm from Lebanon, so olive oil is pretty much like water for us. Let me ask you a question that kind of tries to join some of your world and mine. So I work in the area of evolutionary psychology. I specifically apply evolutionary psychology in studying human behavior in general, consumer behavior in particular. And one of the things that I often explain to people, to explain to them the value of an evolutionary lens, is the distinction between approximate explanation and an ultimate explanation. Are you familiar with these terms?
I am.
Fyodor? Yeah. So let me explain for maybe some of our viewers who may not know.
Please.
So approximate explanation is what most scientists do. It's the how and the what of something, explaining how the mechanistic explanation of the phenomenon. The ultimate explanation is the Darwinian why. So for example, I could study pregnancy sickness from a proximate perspective. How do changes in a woman's hormones affect the severity of her pregnancy sickness symptoms? But the ultimate explanation is that women have evolved this mechanism because during organogenesis, during the first trimester of their pregnancy, they don't want to be exposed to teratogens, food pathogens that might wreak havoc to the developing embryo, fetus, and therefore it's kind of an insurance policy. So that physiological mechanism, the aversion to certain foods, the attraction towards pickles, for example, serves as kind of an antimicrobial, antipathogenic mechanism. Okay? Now, when I look at CRISPR, I see it as a very mechanistic thing, right? That's beyond my comprehension, but it's in proximate world. But is anything that was resolved in the CRISPR technology, did it come from an evolutionary understanding of things? Or could someone have developed that technology without any knowledge of the ultimate Darwinian why? Do you follow what I'm asking?
I do. Technology development is, as you well know, seldom a linear path. And genome editing in its present-day form is a large quilt with many tiles that have arrived from distinct destinations. And just to give you a brief preview and credit and honor the scientists whose curiosity has brought us here, we need to mention a scientist at the University of Utah named Dana Carroll, who studies how DNA changes bits in frogs. Again, not exactly a cure for sickle cell disease. We need to mention a scientist at Memorial Sloan Kettering named Maria Jason, and she basically studies how DNA damage is repaired. Again, not exactly sickle cell disease. And then, of course, Jennifer Doudna and Emmanuel Charpentier were just recognized with a Nobel Prize for making probably the most important scientific discovery of the past 25 years that gave us CRISPR technology. They did not set out to build an approach to change DNA. They were driven by curiosity about these extraordinary machines from the realm of bacterial defense. I think if I were to take a step back and look at the, let's see, 45-year history of thinking and then trying to change DNA, the overarching theme is DNA of larger things, and I want to be respectful of our colleagues who work on, let's say, yeast, which is small. DNA of larger things really hates to be changed, and the deep evolutionary why has to do with the way that our larger organisms evolve and come together. Smaller things have incredibly plastic genomes. A pathogenic form of E. coli that causes food poisoning. Bacteria have about 6,000 genes. If you take the same bacterium, quote unquote, that is pathogenic, causes food poisoning, and you will see this acquire 2,000 new genes. I'm not kidding. Like literally 30% new genes just because. So that the profound evolutionary tension here, the deep why, is between smaller things that love changing their DNA because they're opportunists. Let's try random things and see what happens. Right. And larger things for whom the inverse is true. In the biggest sense of the word, the invention of editing has been the introduction of tools and ways and means from the smaller things into the larger things.
Nice.
That's beautiful. If I were to chart, like, thank you for us. I've actually never had to think about this before. So this is
cool. That's
what
wonderful.
I can offer.
Okay, let's maybe we'll do another 10 minutes. Is that okay, Fyodor?
How about 10? Exactly 10 would be perfect.
Yeah. Okay. Perfect. So let's discuss other things, kind of out of the not the myopia, but the specific technical things related to CRISPR. Undoubtedly, you receive tons of applications for prospective doctoral students and so on. What makes for a great doctoral student and then eventually a great scientist? And I think you already answered that partly when you were referring to the two Nobel Prize winning ladies when you said curiosity. So I would suspect that just a deep reverence and intellectual curiosity for all things interesting would probably be top on the list. What else would you say are some of the non-negotiable attributes that I must have to be a good scientist?
When John Lennon was asked what is the secret of the Beatles, he said, listen, if we knew, we would form 4 separate bands and manage them. If there was a one-size-fits-all recipe for a successful or accomplished or just a good scientist, there will be multiple books. And I've had the honor of working, let me think about this, with 2 Nobel laureates. And I trained in the lab of one of the most stratospherically brilliant people I've ever met. And currently, I share faculty duties in here at the Innovative Genomics Institute and MCB with just insanely brilliant people. And they're all so different. Right. They really are.
You must have some things that are similar, right? I mean,
well, so and now let's try to draw this very, very large Venn diagram.
Exactly.
They are, I think, all driven by a sense of awe between two things, before two things. The first is the beauty of Mother Nature. I don't know of a single accomplished scientist who doesn't think that nature is beautiful. And you can think about the glory of the rainforest or the exquisite beauty of the structure of DNA. You can think about ecosystem size beauty or beauty at the level of individual molecules. I think this aesthetic awe is a common theme. We are all in aesthetic love with what we study. It's just like, so different people find different things beautiful. So I think that's one. The other is a reverence and inspiration from the fact that all of us are standing on the shoulders of giants.
Yes. Famously stated by Newton first, correct?
It's interesting you should say this, because the two Nobel laureates have had, it gives me goosebumps to even say this, that I've worked with. One is Aaron Klug. He was the president of the Royal Society, and he actually, when I visited him in Cambridge, he honored me with a visit to Trinity, which is, of course, Newton's college. Believe you me, if there are more out-of-body experiences than having Sir Aaron Klug give you a tour of Newton's chambers, it does not get more out of this world. If Paul McCartney gave me a tour of the cavern, which no longer exists in the original, unfortunately, I would be less awed. And it's extraordinary for me to just witness this sense in which every scientist feels themselves connected to several millennia of human intellectual and moral rigor. And again, when Jennifer won the Nobel Prize, there was a small celebration, and Jennifer trained with a Nobel laureate, Jack Szostak, and he said something very affecting. He said, listen, people, the only thing better than winning a Nobel Prize is having your student win the Nobel Prize.
Wow.
I know, we all fainted in the best sense of the word. But I think, I've said a number of occasions what makes Jennifer special. When Newton was asked, how did you discover the laws of motion? He said, by thinking on them continuously.
Right.
Jennifer comes across as a person who, in Newton-like fashion, has thought continuously for her entire life about a specific set of problems. And I think that comes from a sense of belonging to a particular human tradition.
Right.
We are all craftsmen in a particular human realm. And some are much, the contributions could be small, but the sense of honor, of belonging, it gives me goosebumps to say that I even sat in the same room with Aaron Klug or Jennifer Doudna or, Randy Sheckman, a Nobel laureate here in my department. I speak with him, and it's always awesome. It's like in the formal sense of the word. But the sense of honor and pride and belonging to that human tradition is a major driver. Is really.
I love both of your explanations, but your former one where you talked about the aesthetic, I really love that one because so I studied in my undergrad, I studied mathematics, and when you solve a theorem using a language that is impenetrable to 99.9% of the people. I can't go back to some of the stuff that I solved when I was an undergrad and understand it today, although I think I remember most things I've ever studied. And there is truly a reverence for, as you said, I'm not a very religious person, but I always used to say that God speaks through mathematics, because when you understand mathematics and you see some of the beauty of it, it's almost so divine that things can work out that way. I can imagine using bioinformatics, you get the same kind of response when you do some sort of genome analysis and so on. So I really appreciate the answer. I have a conversation that I'll be holding soon with the philosopher A.C. Grayling. I don't know if you know him. And we'll be talking about what is beauty. So maybe I will steal your former answer as part of my my own answer. Beauty is really being in perpetual awe of nature. So I guess maybe my next question would be, and I'll wrap it up soon, do you think that we can have that kind of aesthetic reverence to the scientific things that we study without us then falling into but therefore this must be God? And I don't mean to put you on the spot by talking about divine things, because I get both people who say you can't be a scientist and not believe in God when you see how exquisite nature works. And of course, the other side says you can't be a scientist and believe in the supernatural. So where do you fall on that dichotomy?
I think the central message that has guided me is actually Stephen Jay Gould, the great late evolutionary biologist. And he wrote...
You're going to go to the NOMA principle.
Close. He wrote an essay in the New York Times called Leave Darwin Out of It. And the essay is about people who are trying to use evolutionary justification. Oh, you know, humans evolved as hunter-gatherers, and therefore the following set of horrible behaviors are justified. And Darwin says, whatever science does can never decide the morality of morals.
Right.
So who we are as moral beings is not just metabiological, it is separate from biology. You know, we as a species have spent 200,000 years evolving in all sorts of ways. We domesticated the dog 35,000 years ago, and rice 10,000 years ago, and the cow 5,000 years ago, and then all sorts of things happened, and our growing appreciation and understanding of what a moral, ethical life is has nothing to do with what we understand about the molecular makeup of living things. And that is my central principle in thinking about this. I think it was Voltaire, who famously said when asked where is God, or maybe Laplace, where is God in the model of the universe? And that person said, I had no need for the God hypothesis, right? A practicing molecular biologist has no need for God either to be in awe of nature or to study it. Is there a divine entity that implanted the concept of morality in human brains? I have no idea, but it doesn't matter.
Right.
Being a moral being is what matters. And whatever the origins of that, we have a duty to that, irrespective of its source. In other words, I do not think that saying that the first and second law of thermodynamics are true, that, you know, DNA is a double helix and affects living creatures by creating messenger RNA that is translated into protein. All of this atomic level understanding of nature that we now know never was not created. It evolved in Darwinian fashion. The fact that that is true, to me, has no bearing on the morality of morals.
Right.
What is wrong is still
so
wrong.
it is very much in line with Gould, who talked about non-overlapping magisteria. We don't need to put religion and science against one another. They can coexist and everybody will be happy. Nice way to end this conversation. Stay on the line for a second. Fyodor, what a pleasure to have you on. I could talk to you for hours. Thank you so much for accepting the invitation.
Truly a pleasure.
Cheers.